Hey, welcome back to Microbiology. Today, let's talk about bacterial growth. Happens thanks to binary fission, and the cell replicates its DNA, and just replicates the bacteria alongside with it, and there is a thing called exponential growth when everything starts when all the bacteria starts replicating, and the doubling time is going to be equal to the generation time. So the second generation is going to be when the original amount of bacteria doubles. So that's the exponential time. The lag phase is during the cell replication, and the log phase is when the cell division is greater than the cell death. Antibiotics are going to work best during the exponential phase. And the plateau is when the cell death is going to be equal to the division, or when the bacterial cells start to sporulate. And then during the decline or the death phase, that's going to usually happen because the antibiotics are working, or the host immune system is starting to catch on and kill the pathogen. There can be autotrophs where they make their own nutrition, heterotroph when they steal nutrition from the host, or chemotrophs when they use certain chemicals such as sulfur as their source of nutrition. Most pathogenic bacteria are heterotroph, which makes sense, and they need the essential elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfate. Or sulfur. This is chomps. They also need some minerals that the host provides, and the organic growth factors, which are going to be the vitamins B2, B1, the nicotinic acid, B6, B9, and B12. Okay, so the obligate anaerobes need air to live. They're going to grow at the top of the tube, and as you can see here, the obligate anaerobes are going to grow at the bottom of the tube, furthest away from the oxygen, and they form like crystals in the tube. Facultative anaerobes, that means they don't really need oxygen, but they can. They work best with oxygen, so it's going to be more towards the middle top, somewhat dispersed in the tubes. And the aerotolerant anaerobes, they don't need oxygen, but the oxygen isn't going to kill it, so it's going to grow out throughout the tube, making the entire thing cloudy. And the microaerophiles are going to be the bacteria that requires some oxygen, but not a whole lot of it. So they kind of grow towards the top-ish middle of the tube. Obligate anaerobes lack the superoxide dimutase and the catalase or peroxidase to neutralize the reactive oxygen species. So they undergo the lethal oxidations when exposed to oxygen. Capnophiles need carbon dioxide, and the saccharophiles need cold temperature. Mesophiles, which are the majority of the pathogens who infect the human body, need a moderate temperature, and the phototrophs are going to be the ones that need sunlight, but that's not really important. What's more important is the saccharoph, the mesotroph, and the thermal phials, or the hyperthermophiles. These don't really pertain to human infections, because this is more towards the deep sea vents and stuff. Okay, so the culture medium is a material that allows the bacteria to grow and replicate on it. It's usually agar. A pure culture is going to be only one type of bacteria grown. A colony is going to be like this. It's a growth of a visible mass. The basic media is going to be your basal media. It requires the bare minimum. The enriched media is going to be special media to help grow certain bacteria or help grow them faster. So These are going to be blood agar, this comes from sheep, or chocolate agar. Chocolate agar is not made out of chocolate, it's actually made out of sheep's blood, and the red blood cells in it is going to be lysed, or the hemoglobin is going to be oxidized, making it brown. The selective media only allows the growth of the one desired microbe, and the differential media is going to be used to distinguish within like a bunch of different organisms. So they can be the McConkey agar or the EMB agar. So the EMB is eosin methylene blue, and the McConkey agar to identify the G bacteria. So the McConkey agar is when lactose fermenting colony make it pink, and the non-lactose company make the agar yellow. The EMB is going to form like a metallic green colony if it is lactose fermenting. So it's really to identify the gram-negative bacteria. And the MSA, mannitol salt agar, is going to isolate the Staph aureus. So the Staph aureus is going to turn the agar yellow, and that means like the fermentation is positive. So excuse me. So the gram negatives are going to form lactose. Sterilization is going to kill every single form of microbial along with the spores. So this is going to be like nuking everything. Disinfection only kills the pathogenic microorganisms, so spores are left alone. Antiseptics are going to be allowed to, they're going to allow you to use it to the tissue, unlike the disinfectants, and they inhibit the growth of the microorganisms. The disinfectants or the sterilizers are too harsh. It's going to kill healthy tissue. You can use the physical, mechanical, or chemical means in order to sterilize and disinfect. Okay, so heat. Basic process, it's going to denature the protein. So in dry heat, we use incineration or direct flaming in order to sterilize like heat-resistant materials for immediate use. So this can be surgical tools, such as scalpels. So these are your inoculating loop or a hot air oven. Moist heat is going to work by coagulating and denaturing the enzymes and proteins. So we use it in pasteurization, which can be it can be accelerated in flash pasteurization and ultra pasteurization. So in these, you shorten the time and increase the heat. Boiling kills a lot of pathogens, but it won't kill spores, so it's not really immune of sterilization. It's immune of disinfection, but not sterilization. Tyndallization is boiling over a bunch of days, and it will manage to kill some spores. Not all of them, but some. Autoclaving is going to be the most useful. This thing is going to be steam under pressure, under a lot of heat and a lot of pressure. This is going to even destroy the prions. So this is the basic mechanism of an autoclave, and it's going to be mostly used in the hospital. So we're going to use this to disinfect, or not just disinfect, sterilize the majority of your your tools. Okay, so radiation, UV radiation destroys the DNA of bacteria by making the thymidine bonds, and it's mostly used to sterilize the air, the operating room. So it's going to be sterilizing the surface, the cabinets. It also sterilizes the public water supply. Ionizing, these are your X-rays and gamma rays. These are better than the UV because it has higher energy and greater penetrating power. But it can create oxygen and hydrogen radicals from water. So this can be good in order to sterilize, but it also is dangerous to humans. The process of creating the reactive oxygen species is to denature the proteins and the DNA. So this is really used to sterilize the heat-sensitive materials. So the nitrile gloves, your syringes. The mechanical means of disinfection is just filtration. You use that to sterilize. No, I'm sorry, it's sterilized. You use that to not really disinfect either, just to maintain, I suppose. You're going to clean. I guess that's it. The heat-sensitive liquids, which are going to include like the enzymes and the solutions for IV fluids. And you can also use it to, it's not really the right word, clean the air in the safety cabinets. Okay, so more importantly, the chemical means. They disrupt the cell membrane, they denature the proteins, or they break up the DNA. And the rate of killing depends on the concentration, of course, of the chemical, the time of exposure, the physiological condition of the microbes, the presence of organic matter, so whether it has been contaminated or not, then the population size. The phenol and the phenolic derivatives, so the carbolic acid, are very good. They dissolve the lipid solvents and they disrupt the cell membrane. It's actually used by Dr. Lister, the father of modern surgery, to be like the first antiseptic. I want to say he was in England, but he was the first to like really make it mandatory for anybody going into the OR to disinfect themselves using carbolic acid along with disinfecting the patient. However, it's very toxic. Nurses and surgeons were getting their hands burnt off, and it's not sporicidal. So that actually led to the new invention, surgical gloves. So, where there's a need, you fill the need. Maybe sometimes, I don't know, somebody's going to be able to make a antiseptic that's not quite as damaging to the tissue in the future. Alcohols, ethanols, and isopropanols are going to dissolve the lipids and denature the proteins. They're not very effective against spores and non-encapsulated viruses, but it's good enough to use in like a daily at-home like countertop. They're most effective at 70% because of the properties allowing it to dissolve the lipids and whatnot. It's most effective with the water, like with some water content. Aldehydes are going to denature proteins and modify nucleic acids. So we use this glutaraldehyde in a 2% sterilant solution for heat-sensitive instruments. The problem with aldehydes for human use is it is carcinogenic. So formaldehyde, that's the other one. Formalin is the new formula. It's about 37% of formaldehyde in aqueous solution, so the carcinogenicity is decreased. Halogens are going to be gases, and those denature the proteins by oxidizing them. Chlorine will purify water and disinfect. Iodine, we use this in surgery to prep for surgery, for incisions. So this is going to be it's going to sterilize, not really sterilize, disinfect the skin. Oh, and it's antiseptic, so very good in wound care as well. Heavy metals are going to denature proteins. We give sodium nitrite to prevent eye infection in babies born from moms with gonococcal infection. And hydrogen peroxide is an oxidizing agent which attack the sulfidyl groups in the proteins, so it's going to attack the protein the proteins in the spore cell wall, so it's a very good antiseptic. Ethylene oxide is a gas that's bactericidal even against spores, so that's pretty good. It works by alkylating the proteins and the nucleic acids. It has a great penetrating power, so it's going to sterilize like your bedding, your heat sensitive materials. Can't use it on humans though because it's very toxic, carcinogenic. Anything that acts on the nucleic acid is carcinogenic. Detergents, which are your quaternary ammonium salts, your benzalkonium chloride, which is also known as Lysol. These are strong surfactants. They disrupt the cell membrane, and they're very effective against gram-negative bacteria and the enveloped viruses. Of course, because the enveloped viruses have an envelope made out of lipids, right? Bleach is a pretty good disinfectant. It's effective against a lot of, bacteria, along with the hepatitis B or C virus. Okay, so the lab diagnosis, we first used the phenotypic method to observe the characters. So we're going to try to look at the microscopic features, the cultural characteristic, the biochemical characteristic, biotyping, stereotyping, and the sensitivity to antibiotics, one of the more important ones. Immunological, it's going to be in vitro using serology, or in vivo, the PPD test for tuberculosis. And there can be genotypic tests, which is like the fish assay, not very commonly used, is expensive and time consuming. So the phenotypic method, so it's just like, using your senses, compare and contrast. We want to see the shape, the size, the staining, the reactions, if any, in the cell structure. We want to look at the colony appearance. Is it like little dispersed colonies? Are they all grouped together? And the biochemical process or characteristics. So if there's a presence or absence of particular enzymes or metabolic paths. And the antigenic, we want to use serotyping, and that's important to find the sensitivity to the antibiotics. So in the microscopes, we want to observe the shape. We use the dark field to look for spirochetes, so syphilis. For the compound or bright cell, we're going to observe the host cells that are infected, along with any kind of other microorganism. In the phase contrast, there's going to be analysis of the internal structure of the living cells. We want to see, like, the DNA has been, or the nucleus has been affected. We want to see if the mitochondria is okay, Golgi apparatus. Use the UV or fluorescent staining for fast diagnosis. You want to examine the CSF, tissue biopsies, and the cultures, as you can see here. And you use the electron microscope to really study virus. We don't use the electron microscope to diagnose at all. It's very expensive and it takes forever to do. So we use a primary stain and a counter stain in a gram staining or an acid-fast staining to see an endospore. In a negative staining, we use India ink to stain the capsule, so it's going to be on a dark background. There is one dye in a simple stains. We use that to see spores. It's pretty straightforward. Gram staining, there is first the primary stain and then a counter stain. And the acid fast is going to be your Ziehl-Neelsen staining. So the Ziehl-Neelsen staining, it's somewhat the same. You want to stain it first with something, usually some kind of fuchsin stain, and then you use heat to adhere the cells to the slide and decolorize it with some kind of acid or alcohol, and then a counter stain of methylene blue. Okay, so when analyzing a culture, you want to do a streak plate, and from there we see a clean streak is going to be a pure culture, and if there's going to be isolated colonies, it's contaminated. So it's useful for the biotyping for specific enzymes, the toxins, and the use of specific nutrients because of which culture is used, right? For example, E. coli uses acetate as the sole source of carbon. So the agar might be like a specialized agar that's impregnated with a lot of acetate. You're going to use the culture to identify the colony characteristics, the antibiotic sensitivity test is very useful using this. And as you can see here, here's all the streaks, and we're going to use an optochin test using the optochin disc and S viridins. Let's see if there's a resistance to the antibiotic impregnated in the agar. So in A, there is going to be a lot of growth, as you can see here. Like, it's a very thick, opaque layer of growth, like this heavy confluent growth, that's what it's called. And there is a resistance to the antibiotic. On the other hand, this particular pathogen, S. pneumoniae, is going to be susceptible. There's much less growth. So that's our sensitivity test. A catalase test, we want to see if anything bubbles up. We're going to see if hydrogen peroxide is made. Carbohydrate fermentation test is basically a pH test. We want to see if there's carbohydrate being used up, there's going to be a change in color, and it's going to produce some kind of acid and gas. In the oxidase test, it's going to detect the aerobic pathogens, and in lactose formation, that's what we test for. It's somewhat the same, right? So we want to see if anything bubbles up. Stereotyping is going to be used to identify the isolate or detect any expressed antigen. Coombs test, so agglutination to identify the antigens that's going to be present on the specific microorganisms. We look for the IgM and IgG levels. If there's high IgM levels, infection just started. Higher IgG levels, infection is starting to resolve, the immune system is kicking in. We use PCR hybridization and sequence analysis in order to really identify the antigens and antibodies. So that's it for now. Thank you for your attention, and good luck studying.